G'day, good morning. Thank you for listening. This is an agricultural show, and I respect the law, and I hope you do too. Today's information is for educational and scientific purposes. The websites or papers that have been discussed will be put in the description below. And on today's topic, it could be an open topic for questions, or it might be up to the soil science and fertilizers course that I'll be going through. And I think that's up to week 5 or 6. It's up to the macronutrients. So I'll see what chat dictates to how the show's going to be titled today. So I'll give it another minute. So if you're rewatching this back, fast forward till you see some slides use a slide bar across the bottom, and then you'll see when the show actually starts. And there is a show today, because usually when there's no chat or discussion or questions, I have an early day, and there's no show for the week. But because I'm going through this soil science and fertilizers course, university course, we've got some action, at least for the next 6 weeks, so I can get to the end of it. If there's no real questions or discussion topics, so I'll give it another 20 seconds till 2 minutes, then I'll start. Give the folk enough time, because there's a 30-second delay between when the words are written to when I see it. So just in case there's been some questions or discussion points put up, that's why I'll usually give it a couple of minutes until I get into things, which is right now. It's 2 minutes. So hello to Mace. Hello to 420 Fred. So if you've got any questions, put them in now, because otherwise I'm going to start with, let's have a look. Week 5, micronutrients and their role in plant nutrition. So there's another 30 seconds. Doesn't look like there's much coming through. So I'll start sharing the screen and getting into the course. This one. So I've already done all these and testing. I thought today was up to manures. I can't wait till it's up to the organic bit. All these stuff, I can really elaborate on that. Soil health and quality. That's substrate health and quality too. But this is what we're on to today. All right, let's start. So soil health indicators. So today's topic is week 5, micronutrients and the role in plant nutrition. So soil health indicators are macronutrients, micronutrients, physical condition, biological health, and other abiotic stresses. So they're the things you want to keep in check. Micronutrients, what is it? Well, micronutrients represents some essential nutrients that are required in very small quantities for the growth of plants and microbes. There are 17 essential elements for plant growth. And out of these, 8 elements are considered the micronutrients. There's iron, manganese, copper, zinc, boron, molybdenum, nickel, and chlorine. And out of these micronutrients, some behave like an anion, and that's the iron, manganese, copper, zinc. They behave like cations, and then the ones that behave like anions are boron, molybdenum, and chlorine. Well, it's because they got pluses next to them, and they're cations, and they've got minuses in their element. Essential plant nutrients, so the macros, and then the micros. So I've already been through all the NPK, CalMag, sulfur, and then today is into the microbes, micronutrients. So that the chemistry of micronutrient cations are different from micronutrient anions. Among these 8 micronutrients, the content of iron in soil, as well as plants in the highest and sometimes higher than even phosphorus and sulfur content. Micronutrients are also called trace elements, oligo elements, or spur elements. Why is it micro? Well, the uptake is low. It's measured in like this chart will show us. No, I want the percentages, 1.5% and stuff like that. Really, I'll scroll through a bit because it's in here. Uh-oh. Maybe it's not in here. It's not. I'm going to find it because it's pretty important for what I was going to say. Might even be in here. No, it's not. Fair income. Really, truly, it should be. Oh, it's this one. I'll just go through and be in this one. I wanted to show the percents of it, how low it is, why they're called micronutrients. There it is. So you've got your three main ones. This is all your essential elements. So 3 out of the air, and the NPK, cal, mag, sulfur, and they say silicon's in there now. But see how that's in a ppm or a percent amount? Sorry, this is in a particles per million is the micronutrients because they're in such little amounts, and the macronutrients are measured in percents. So that's what I wanted to get to. So these ones are going through today. They're so little, and look at molybdenum, it's 0.1%. But if you don't have molybdenum, like in some Australian soils, nitrogen struggles to function because it is a cofactor. But the whole reason for this is just to show the chart to say how low of an element they are needed, but they're extremely essential to require for the completion of a plant's growth and development. Lieberg's law of the minimum, been through that. So the critical information in plants, that's when they're discovered. The average concentration in plant tissue, I kind of just went through that. Why micronutrients is a matter of concern. Well, within the last few years, the nutrient reservoirs in soils were gradually exhausted, and it's no longer possible to sustain higher yields even by applying nitrogen and phosphorus fertilizers. And one of those reasons was the molybdenum in Australia. Go on to read, micronutrients deficiency are observed in many crops. Okay. They first noticed that zinc was removed in rice. Okay. I'm not going to heavily go into it. I'm just going to briefly touch on it. What does this represent? The impact of the green revolution on the emergence of micronutrient deficiencies. Yeah. Well, in 1960, nickel was added, and they've fiddled around with it in the last 10 years. Even they've gone up to 19 and back to 17 because they've realized a few elements were beneficial, not essential. But this is an impact as they've found of the micronutrient deficiencies, so they're still going up and up. Most important micronutrients in Indian soils are zinc, boron, and molybdenum. Molybdenum's very important in Australia because it's lacking. It's been removed. Functions of micronutrients. Well, they reckon borons in sugar translocation, chlorines in stomata opening, copper accelerates photosynthesis, iron helps in chlorophyll formation, manganese acts as a autocatalyst, molybdenum is nitrogen fixation and a stimulation element, zinc promotes growth hormones. See, by having these at a level that is required from the plant in your substrate, these functions can be performed successfully in the plant without any stresses. Then it's going to go on to say how each one works. Zinc's an essential component of 3 enzymes, and it's those enzymes are good for various processes. Zinc's important in the synthesis of indole abscisic acid, auxin, a rooting hormone. Essentially, it's a rooting hormone, and essential for water uptake. Zinc is, and it plays an important role in the stabilization of protein. What does iron do? Well, iron's important for 2 groups, the heme protein and FES protein. Did you know in meat, when you crave meat, you crave heme? Isn't that interesting? Iron's major function is involved in the production of chlorophyll. It's also an oxygen carrier, nucleic acid metabolization, chlorophyll synthesis, protein synthesis, acts as a catalyst in nitrogen reductase, structural component of porphyrin molecules, and it's required for nitrogen fixation. Iron is. Boron is a primary function related to cell wall formation. It's important for pollination and pollen germination. So if you're into breeding, you want to make sure you have boron at a good level. It helps in potassium translocation as well as somatal opening, calcium metabolization. Boron acts as a regulator for potassium-calcium ratio, and it's essential for the translocation of sugars. Here's some little graphics of how boron helps. Boron is the key to high crop productivity because especially for dicots, which need pollinization. Monocots produce female and male, so they don't really need it. They can self-pollinate. Copper, what's that good for? It's necessary for carbohydrate and nitrogen metabolization. Copper is an electron carrier in oxidation reduction reactions. Copper utilization of iron in chlorophyll synthesis. It enhances the fertility of male flowers, and it enhances disease resistance in plants. You might see copper used as a fungicide. Manganese is necessary in photosynthesis, nitrogen metabolization, and to form other components required for plant metabolization. Manganese is an important constituent of superoxide dismutase, which is a very, very important enzyme for reducing reactive oxygen species. Manganese takes part in electron transport in photosystem 2. Manganese acts as enzyme activation, and this enzyme produces amino acids. Molybdenum is an important constituent of nitrogenase enzyme, which helps in nitrogen fixation of legume crops. Actually, well, wherever nitrogen fixation is involved, so if you're using nitrogen microbes, it's also a process that is required. Molybdenum is an essential component of nitrate reductase enzymes. It's also protein synthesis and sulfur metabolization are affected by molybdenum. Molybdenum has significant effect on pollen formation, also reported to have an essential role in iron absorption and translocation in plants. Possibly that's maybe why Australia has a lot of deserts, because the pollen formation has needed in plants has been exhausted because molybdenum isn't there. Chlorine functions are essential for stomata opening. Chlorine is related to electrical charge balance in physiological functions in plants, and it indirectly affects plant growth by stomatal regulation by water loss. Very important in Australia or very hot regions. I'll just stop here for a second, see if there's anyone to say good day to. Oh, there's Gear Changer. G'day, Gear Changer. Morning from the Gold Coast, he says. That's very nice. Out and about, mate. Very good. Just going through this soil science and fertility fertilizer course. All right. Next lecture. Nutrients in soil and their major roles, and their major carriers. mineral source, name of the salt it carries. Oh, it's okay, and it's micronutrient. All right, that's what these micronutrients can get mixed with to hold the element. What's that start at the top right? Major carriers. Okay, very good. Micronutrient mainly exists in 5 fractions of soil: organically bound, exchangeable, complex, absorbed, or chelated. 4th is oculated and bound in carbonates, sesquioxides, and secondary minerals. And the 5th one's primary minerals. That's how it is. And the cycle through the soil, well, pretty much how does the minerals get exchanged? Absorption, desorption, from precipitation and dissolution, organic matter is exchanged, and rhizosphere processes. Effects of soil pH on micronutrients. Well, soil pH affects availability of micronutrients in general. The solubility and the availability of micronutrients are the greatest in acid soils and lowest in high pH calcareous soils. Exception is molybdenum. In some soils, high levels of soluble iron, aluminium, and manganese may be toxic to plants. You don't want them aluminium anyway. It hogs the cation exchange as the highest bonding strength. Effects of soil pH on micronutrients. In well-drained, aerated, and oxygenated soils, pH controls the availability of iron and manganese. And here's a good chart of the pH effects of plant nutrients. So this is why you want to be between the sort of 6.2 to 7.3 range, because you can see all of these elements are green. So that's why if you've got a iron, manganese, boron, or copper and zinc deficiency, you might think, a toxicity, you know that your pH is getting too low because they're becoming too readily available, and all the ones the plant requires, mostly the primary nutrients, are becoming unavailable. See the colors goes down. Look at phosphorus. If you go to a 6, it's red and struggling. So there's not much, even if there's heaps in your substrate, there's not much available to the plant because of the pH only. That's why pH is so important. Mobility of micronutrients in plants. Well, the mobile ones, where the symptoms appear in older tissue first, are chlorine and molybdenum, and the relatively immobile ones are the metals, which appear in the younger tissues: the iron, zinc, copper, manganese, boron, and nickel. Mobility of micronutrients in soils. See the difference in the plants to the soils? So which ones are mobile in the soils? Well, they're leachable. So you've got to be careful with the chlorine and boron. And relatively immobile is zinc, iron, copper, manganese, and molybdenum. So it's very similar. Soil organic matter is so important. It's an important source of micronutrients. It has higher content of active, non-lignified soil organic matter, implies higher micronutrient availability. It's simple organic compounds as chelates. Remember, chela is Latin for chlor, means it locks onto and holds nutrients together. Like it in the top right, you've got your metal iron. You can use your EDTA, which is a chelating formation, which ends up in a chelation and holding on to that nutrient, that metal or the iron, called ligand. Chelated forms of zinc, nickel, and copper can increase the concentrations of each metal in soil solution to increase their diffusion to the roots for plant uptake. Sulfur, zinc, and boron deficiencies are more likely to occur in soils in low soil organic matter. So even grass clippings is good. If you get good grass clippings without any seed in it, you shouldn't have to put it through a thermophilic process to get it up to its temperature to reduce the seed content. You're just not killing all the pathogens off. That's by not putting it into that. But it has the correct carbon to nitrogen ratio. So you just get your grass clippings, put them in a heap, and use them at will. And there's a great source of organic matter. Soil organic matter deficiencies in copper and manganese are most common in peat soils. Iron, copper, and manganese are more available under waterlogged than aerated conditions. And in well-drained soils, pH controls the availability of iron and manganese. Organic chelation happens with many organic chelates have not been identified. However, components such as citric and oxalic acids form during decomposition, having chelating properties. Oxalic acids, really? Huh? Oxalic acids is produced from red sorrel, and it's not good for animals to ingest because of the oxalic acids. So that might be good to use as a chelating property. Cycling of chelated micronutrients. Okay. Goes out through the root tip, comes back through the root hairs, similar to the rhizophagy scale cycle. What's it skipping for? I'll press it. Terms used to describe levels of nutrients in plants: deficient, critical range, sufficient, and excessive. So deficient is low enough to severity limit the yield. Critical is well below in response where you might get problems. Sufficient is optimal. Excessive is too much. So you're starting to be adverse effects from loading up too much. And then you can also get saline conditions if you have too much as well. And that salinity is not very good. Typical dose response curves for essential elements, macros and micros. Essential combination, element concentration. What's in this y-axis is the plant yields. So as you go in severely deficient, hidden or hunger, and then to optimal, and it goes the opposite way into the toxic. So it's kind of just a graph of explaining what those elements, what those writings meant before. It's a nice, pretty looking slide. Shame it's blurred. Micronutrients in plant at adequate levels. Also, it just tells you how many milligrams per kilo is required for plant. So in deficiency levels, it's under 10 milligrams, for instance, in boron, and in toxicity levels, it's probably over 10 or 20. It's hard, I couldn't read it, sorry. That's what that slide depicts. Pretty cool. Good to have the numbers. I like the numbers. How to identify deficiencies in soils? Well, in plants, it's easy to identify because you can see the leaves. But in soils, soil is an element where it travels through the plants, so you can use the plant as a identification purpose of the soils. Usually what happens above the ground is a good indication of what's going on below, because with for instance, red sorrel, that's only likes acidic soils, or blackberries, they like acidic soils. So if you see those two growing, you know that generally the pH below is going to be in acidic levels. What else can I pull out of this slide? Depletion in metal ions. Okay. Depletion in free metal ions due to the formation of DTPA complexes. Okay. Not really helping. Average removal of micronutrients by different cropping systems. Here's some examples on how through a heart growth and development, and then you harvest, what's removed from the soil. These are just some different examples of that. That's why adding organic matter, which pretty much has all of this stuff in it, is a fantastic source after you've cropped out and you're tilling again, or if you want to top dress. Causes of micronutrient deficiency. What are they? Well, it can be the parent material, high yielding or hybrid varieties, imbalanced nutrition applications, soil pH, soil condition, low organic matter, over liming, micronutrient free, high analyzed fertilizer, okay, land leveling and scraping, or interactions between nutrients. Why is it skipped? Micronutrients of little concern. Well, I reckon this slide says calcium, magnesium, manganese, and copper. Reasons for deficiency: very acidic soils, rare deficiency. So magnesium is a very acidic, sandy soil. Reasons for deficiency: manganese, high pH, high soil organic matter. Copper, high organic matter, highly weathered soils. Some reasons for a deficiency. So if you see any of these deficient leaves recognized, you might want to go over here and relate to the reasons and see if these are true. More a pH thing. Deficiency symptoms of micronutrients. Well, boron is the terminal growth, shows rosetting. Chlorine, chlorosis, or yellowing in younger leaves. Copper, leaf tips become white. Well, anyway, that's what it says. Iron, interveinal chlorosis. And manganese, interveinal chlorosis in the younger leaves. There's a good a difference. Older leaves, it'll be iron. Younger leaves, it'll be manganese. Molybdenum is when leaves become pale in general. Zinc development is of chlorotic midribs. So the midrib veins in the leaves, you'll see it's going yellow. And remember that leaves go pale is because of the nitrogen, and that's because molybdenum can't synthesize with it. Soil zinc is essential for enzyme systems. See in the slide picture over here, plants are happy with their seeds are galvanized or soaked in zinc, untreated and treated. Essential for, remember that's getting into your nanoparticles in agriculture, which is another fantastic course that I've talked about a lot. Essential for enzymes in zinc, soil zinc. Quite immobile, becomes deficient in flooded soils, may be toxic in problem soils for sustainable plants. Zinc deficiencies. I'm going to go through them because there's quite a few. Phosphorus and zinc. Excessive concentrations of phosphorus in the soil root result in binding of zinc with root cells. Large amounts of starter applied phosphorus can enhance zinc deficiency in soil. Zinc is low and no zinc fertilizer is applied. So how's we going to go through and read this? So that means that's applied, none's applied, the yield is good. The amount in the plant tissue, phosphorus is under 1%. Zinc, ppm, see that's measured differently because it's a micronutrient. So it's just showing the inputs to what you get for the outputs. Iron, let's see if there's any written here. Oh yeah, Aussie Jeffro. Screen has gone black bud for me. I'll listen. All good, I reloaded. Oh, okay. Thanks for the message. Aussie Jeffro. Hope you're doing well, mate. Kind of halfway through this, I reckon. Iron in the plant catalyzes the production of chlorophyll and is involved in several enzyme systems. Deficiency symptoms is yellow and white leaf color. For iron, symptoms first appear in the younger leaves, wider susceptibility in different crops. How to identify deficiencies in plants? Well, this is the leaves now. So this is really cool. So irons, interveinal chlorosis, as you can see in here. Manganese, where is that? Pale green, grey spicks down here. In those ferns. I probably should read all these. Alkaline disease, entire plants become chlorotic for iron because it can't. I'm going to say why. Manganese, pale yellowish green interveinal chlorosis spreads from the tip to the leaf base. It's grey spicks in oats. All right, different crops have different Symptoms. Zinc. Leaf base of younger leaves become chlorotic brown and blotches, streaks on lower leaves. So blotches or streaks, you could think that you have a disease or something like that. Well, it could just be zinc. Copper, the leaves develop chlorotic streaks on either side of the midrib. Where's the copper one? So you know the midrib, that's in the middle. So you just look for either sides of it. There's also dark brown necrotic lesions on their leaf margins or the tips. Nickel has leaf chlorosis and leaf tip necrosis in cowpea. Seen there. Boron is white and rolled leaf tips of the younger leaves. Death of growing points, but new tillers continue to emerge during severe deficiency. No good examples there. Visual examples. Chlorine is wilting, twisting and curling of leaves. Molybdenum in plants. Where's chlorine? There's chlorine there. Hard to see, sorry. Sort of looks like it's wilting even, doesn't it? It's chlorine. So in plants with reticulate venation, deficiency first occurs as chlorotic mottling between the veins on old and middle leaves all over the structure. So you can see here and molybdenum down the bottom. So the older leaves are happening first. There's a more of a exaggerated version in the top right. You could even think it's potassium. Iron deficiency. Boron in cauliflower. See the brown in cauliflower? I thought that was from fungus, but that could be for some growers because I cut that out when I go to eat it. Vertical splitting in carrots is due to boron deficiency. I actually had some of that. See, and that's why organic matter would fix it. Groundnut, hollow hearts, they go hollow from boron. Boron in tomatoes. That's a good source of boron, your borax. It's very close to boron, so it's quite cheap. If you have any problems, don't use much organic matter. Molybdenum, susceptible or tolerant plants to micronutrients and conditions conducive to micronutrient deficiencies. So micronutrient, plants most commonly of deficiency, plants rarely deficient, soil conditions associated with deficiency. So see how this goes into heavy detail and it crisscrosses all the time? So this has already been touched on and a little bit, so I'm not going to go into it too much. Status of micronutrient deficiencies in India. You can see they're horrible everywhere. It's the same as Australia. It's really bad because it's the amount of organic matter also for it to be broken down, which is immobilized, and it can be mobilized by microbes to create that element. And if you've got mostly desert region, you've got no chance in that happening and replenishing nutrient cycling. That's why Australia is horrible, really, really bad. But you still can work with it, but it's just hard. Need lots of organic matter. The status of micronutrient deficiencies in India. It's a combination of 2 or 3 micronutrient deficiency statuses. Different states in India. Becomes basic. I'll summarize again. Boron in soil sources. Oh yeah, how do you get your boron? Well, borax is the easiest way out of all of that because you can nearly buy it over. You can. I can't remember what else people use that for, but it's pretty common. Boron in soil. Total boron concentration in soil depends on parent material and the degree of weathering, with natural background concentrations usually ranging from 2 to 100 milligrams per kilo. Boron's never found as a single element and is usually combined with oxygen as borates. Boron may be tightly bound in silicate materials to produce insoluble minerals like clays, and and the absorption of oxides and association with organic matter occurs. Boron in soil. The absorption of boron, second line, in soils is weak, though generally higher in high pH soils above pH of 8 alkaline. The low retention makes boron vulnerable to leaching, and excessive rainfall can result in the loss of it. Boron content in surface soils is so low it's measured in micrograms per gram. That's, what's that? Micro, milli. Milli's 1,000, micro's 3 more zeros, so 1,000,000 times smaller than a gram. Managing boron is difficult. Deficiencies and toxicity range of boron very narrow, so very small quantities is required. That's why getting all these micronutrients, really, organic matter is just the source. They have a really good source, and watch out for any deficiencies, and then it should be sweet. Yeah, why do you get a deficiency? Because you intensively crop, or you just use the soil and don't feed it. Like too many farmers in Australia, they want to spray it and kill the soil because they think that's the good go. Then they want to go and add fertilizer and think it should be all right because they've gone and added urea, which is basically just nitrogen only, nothing of the other 16 essential elements. And they think, oh, why isn't it going good? Then they'll go and get pests and disease will move in because it's not done organically. They've removed all the microbes and stripped basically of the goodness out of the soil. I went on a bit too much with that. Why deficiency? Intensive cropping, limited use of organic manures, farmyard manures or compost, and why deficiency? Use of high analysis fertilizers. Yeah. Basically kind of what I said. Iron and aluminium oxides absorb to bring boron deficiency. Silicon properties in soil. Silicon is a major constituent of the Earth's crust. 30%. Isn't that a spin out? The silicon is generally not considered as an essential element, so it's a beneficial element. Silicon is assimilated by plant roots as monosylic acid, and it accumulates in the entire layers between and above the cuticle of the leaves. Waxy cuticles. You know, if you're a waxy cuticle like that, if the leaves look shiny, that means they've developed a drought resistance type thing. Silicon concentrations in plant depends primarily on phylogenetic position of the plants more than on its environment, because silicon concentrations in the soil and the soil solution. The role of silicon in crops, well, basically helps with stresses. It's crucial in cereal crops. It's a quasi-essential element. Yeah, it's essential, not elemental. See how science just changes in 99. And silicon is recognized as an agronomically essential element in many countries like Japan, only recognized in the last 15 years. The silicon components in the soil. Well, it makes up 30% of the Earth's crust, so it's pretty much everywhere, unless it's in sandy or desert regions. Silicon nutrition in rice, I won't go through. Symptoms of silicon deficiency. Lowering starch formation and accumulation will lead to yield reduction. Occurrences of diseases such as blast and brown spots may happen from silicon deficiency. Reduced number of panicles per square meter and reduced percentage of filed grains. You could also see cause of silicon deficiency. Soil parent material contains inadequate quantities. Soluble silicon is continuously leached out, and a long period of intense cultivation without help feeding the soil. So back again, it comes to feeding the soil, organic matter, factors responsible for availability of silicon in rice. I'm not going to go through, but you can pause and read that if you get into this. Rate of application of silicon varies. Soil sample tests will tell you things, but otherwise, in general, you should be pretty right, unless you come to a heavily intense agricultural area which has been cropped out without helping the soil. In other words, someone that doesn't practice organics, you might have to look into that. Strategies to mitigate micronutrient deficiency. Well, it's organic matter. Get that and get the right microbes to cycle and break it down. Don't kill the microbes by adding different ozone or hydroperoxide or different poisons and toxic elements that might reduce it, like copper for fungicide. That'll do. Remedies for micronutrient deficiency or applications through fertilizers, tailoring management practices, organics, or selection of varieties. See how that? Number 3, it covers micronutrient deficiencies. Isn't that amazing? But the big man doesn't want you to do that because he wants you to buy all the fertilizers. He wants you to buy all the herbicides because you've killed everything else and all the other different things you need to make those fertilizers and herbicides take effect. Or just practice organics, which is what I promote massively. It's sustainable. Application with mixed fertilizers. I'm not going to go through that because I don't believe in giving mineralized fertilizers. I believe in giving immobilized fertilizers where the nutrients are bound up in material where it has to be slowly broken down, or called organics. A simple principle: right products, right time, right place, right rate. So the right products is put in the right amount of organic matter and manures, the right time. Well, you've got to time it right because you can't put it in at the start or the end or, that sort of thing. You've got to make sure it's done at the right time for, like, for instance, flowering time. You might put in more sulfur, the right place, keep nutrients where crops can use them. So if you want to, you've got nitrogen deficiency, you think, I'll put it on top. No, it'll probably volatiles off. You have to bury it. The right rate, match amount of fertilizer to the crop needs. Definitely, because you can get toxic levels and you can make elements that are power over other ones. 2 philosophies of micronutrient fertilization: insurance and prescription. Insurance means prophylactic measure, a maintenance schedule. Prescription based on soil, plant analysis, prevention, unnecessary application. Methods of application: 3 ways. Well, you can put it to the soil, top dress, put in when you're tilling. You can seed soak or put it on your seeds or dip the cuttings, and you can foliar spray. All these ways are extremely beneficial, and foliar is great for depleting one micronutrient. So if you might see that you're getting some copper, for instance, deficiencies, you might want to put a 50 or 100 ppm foliar spray, mix that up and apply it. And being copper an immobile element, you probably want to see, anyway, been through that copper. Advantages of foliar spray: their rates lower than soil applications, heaps lower. Their uniform distribution, because you can coat the leaves. And foliar spray advantages are response, almost immediate. So cool. You can see within about 3 to 4 hours of nitrogen deficiency. The stuff takes it so fast. And if you're foliating, remember, do the undersides of leaves when you're doing for an IPM spray. That's where a lot of the bugs and problems sit underneath the leaves. But you want to be careful foliar spraying underneath the leaves if something like calcium, because it's very binding, gummy sort of type material, which is going to clog your stomata. Disadvantages of foliar spraying: well, if you put too much on, you'll get leaf burn. Nutrients demand is high when the plants are small, then the leaf surface is insufficient for foliar application. So it's not very good spraying seedlings. Well, it still can help, but it's not as useful. Another disadvantage of foliar spraying is maximum yields may not be possible if spraying is delayed until deficient symptoms appear. You can go through that hidden hunger and then it'll be in deficiency. So you might have had that problem quite some time before you've actually fixed it. And another disadvantage, there is little residual effect from foliar sprays. Application costs are higher if more than one spray is needed, unless they can be combined with pesticides. No, use IPM. If you're going to do a pesticide, it's a biopesticide spray in my eyes. You're going to be putting in your Bacillus, metahiziums, all sorts. I'm not talking about microbes. Some micronutrient fertilizers with nutrient content. Borax has 10% boron in it. It's about all I'm going to, because the other ones are mineral fertilizers. Commonly used boron fertilizer. See, borax. Boronated NPK. Fortified fertilizers, approved in India. Okay. So they've just been chelated and actually I'm not talking about them. Methods and rate of boron application. So how much would you put in of borax if you've got a deficiency? Well, you might put in 1 kilo per hectare for soil, but if you foliar spray, it's only 0.2 kilos per hectare. Isn't that a big difference? So you can see the saving with the foliar sprays. Management practice of soil boron, liming, timing, and manuring. Due to poor spatial distribution of boron, bulk blends containing bulk salts are more effective. No, that's mineral fertilizer. Why more mineralized fertilizer? Fertilizer sources of iron. Just read this. My screen has gone all good. Oh yeah. G'day Blinky, how you going, Blinky? You have any questions? Drop them in chat, please. I'm just going through week 5 micronutrients of the soil science and soil fertilizers course, university course. Written in 2022, so you can pretty much rely on all these slides. Sources of fertilizer, sources of iron. Iron quickly converts to unavailable form. Yeah, you might want to chelate it with the EDHA. Foliar application proves to be most promising for iron. Factors affecting iron bioavailability. Well, it might be high soil pH. Might be too high salt content, salinity. Could be too cold. Poor drainage and aeration. High concentration of nitrate inhibit conversion of the Fe of ferric to ferrous, increasing severity of iron chlorosis. That's how an example of that Mulder's antagonism chart comes into effect, where one element can have an effect on another. Chlorine is soluble, mobile anion with negative. Deficiencies are more likely in higher rainfall areas with no potassium application history. The application of potassium chloride increases yields, reduces incidence of plant diseases. Internal water relationships, osmotic relation, enzyme activation, and other plant processes. Important soil test methods for micronutrients. So, element, extractant, soil solution ratio, shaking period in minutes, and critical levels of efficiency. So it shows on the left here all the different elements. So zinc, a soil solution ratio might be 1:2. You want to shake it for a long time, probably an hour. Put it on one of those shakers, little beaker things. Critical levels of efficiency. It's 1 or 2 milligrams per kilo. So this is more so if you want to test it. strategies for alleviating micronutrient deficiencies, addressing so it's insufficient availability, reduced acquisition, and limited soil content. So application of microbial biofertilizers, so you can increase availability, producing cytophores, that's ion chelators, rhizosphere acidification, microbial redox cycling. Increased, is there any point in reading these ones? It's less, chart down the bottom, lessons in biofertilizer from natural ecosystems. Well, the efficiency of biofertilizers will differ among microbial function groups, depending on the micronutrient in question. Co-applications of biofertilizer with organic matter could be effective in treating some micronutrient deficiencies. Bioprospecting in natural systems is a potential source of microbial taxa capable of mobilizing micronutrients and withstanding harsh environments in the field. Right microbes, right application. That's what that's saying. So they're applying correct sources of micronutrients. Methods. This looks like a summary. Methods of application. I'm not going to read. I don't like the summary thing because I've already been through them all. This is a summary of zinc. In case of orchids, okay, normal soils have 5 to 10 kilos per hectare. Sodic soils, seed treatments, you can dip it in that. Oh, okay, so this is some, if you want to get into your numbers and get down to testing and using zinc, or even zinc as a nanoparticle, these are the amounts that you apply with. And zinc, and you can use zinc as a nanoparticle. Pretty much all of these elements pre-treated with seeds act as NPK fertilizers, was a good summary of my course of nanoparticles in agriculture. So copper and boron, there's some more statistics for that. Some more figures that you might want to throw at. Iron and manganese, different times and amounts to do with that. Same thing again. Look at the iron. Combination of green manures, which is growing green weeds, for instance, or organic manures with foliar spray. So you can even get it out of that, growing weeds and tilling them back through, as long as that hasn't gone to seed. Methods of rate of micronutrient applications of wheat. Recommendations. High yielding varieties, intensive cropping analysis. NPK fertilizers, limited use of organic manures. What? This, oh, that's to do with wheat. Effects of levels of silicon on yield and the quantity of rice. So you can go up to I, because you can see here between how it's reached its level. If they're using 120 kilos, they're getting a 400 kilo output. If they went up to 180 kilos of silicon per hectare, they're still getting virtually the same. So there's always a limit, upper limit and a lower limit for these elements. Effects of applied spraying programs of stabilized fruit fields per plant. I'm not going to go through them. Application of zinc-coated formations and how they work well. That's it. That's the micronutrients. Been through 54 minutes for that already. I tried to rush it too. That's a whole week's of lectures for that. Wowee. Put your questions in now, please. Otherwise, you'll have to wait till next week. Videos, mate. Sort study, here it is. I'm going to show you what we're going to talk about next week. Hope everybody's enjoying the cricket too. I am. Right, now share again. Next week. So we went through that this week. Already done week 6 in soil testing and soil fertility evaluation methods. So next week will be soil health or substrate health and quality problems of your substrate. Land capability classifications also. So yeah, there's quite a few slides to go through. But yeah, soil health and knowing what the problems are and then how to fix it. Gee, it's an asset. It makes it gardening just a pleasure. It really does. That's about it. I'm still sharing. I can stop sharing. And that's it for today. If you'd like to put something in the chat, you're welcome to do it. Put it in right now. And I can put it on the screen. Otherwise, we'll wait till next week. And I'm enjoying this soil science and soil fertility, soil fertilizers. It's a good eye-opener. So if you ever have any problems, you can come back to these slides or to these lectures and just go through and pause it on different stages. That's one of the main reasons I'm putting it up. Because to take all this information in at one point is blooming hard. Like, I just went through a whole week's of lectures. So that's 40 hours worth of lectures I just gave in one lecture, in 1 hour, sorry. So, yeah, don't be surprised if you don't take it all in. A cool thing from my microbiology teacher said years ago that all these names, don't try and learn them. You will learn them as the time goes, like Bacillus thuringiensis. That's good for IPM. And she was right. I used to struggle at the start, and then I thought, all right, I'll take on that attitude. And before you know it, just from using them lots and going through different papers and things lots with organics, they always come up. Then you just learn them, and before you know it, after seeing them and half a dozen times, you know about them. You might want to research about them and find out what they do. So this information will slowly sink in if it's an area of interest for you. What I want to get at, because all this information is so hard to take in. You think, how the hell can I learn all this? Exactly right. I agree with you. But it's a tool to use back over and over. So when you get a problem, you might want to come and rewatch one of these videos. And then you can have the notes in front of you from the slides that are written of recent years, so you can trust them, because science is forever changing. And these ones written in the last few years, you'd hope to rely on them. Yes. Thanks for the nice words, Aussie Jeffro. He says thank you. And says, I appreciate your time spent with us. Good on you, mate. I like the positive feedback. I like negative feedback because it helps me. I call that constructive criticism. It's nice to hear the words. So I do put a bit of time in. There's an hour of my time here. I could have done elsewhere, but no, I enjoy this topic too. Soil science, plant science, and microbiology is just brilliant related to plants. Not humans or anything like that. I don't get into that. Well, they touch on the subjects, but I don't focus on that. I like the plants and the soil, the microbes, and then putting all these experiments to test and to see if they work out. And fascinating enough, they all work out. I'm waiting for something to fail, like plant resistance, and it's so cool. Or gibberellic acid put on to swell fruit. So get yourself a few microbes, which is what I did. And I shouldn't be talking about the microbe bit, but it's about plant hormones. So yeah, I won't continue on talking about that. Jeffro says, I agree. Good on you, mate. Well, we've nearly been an hour, 59 minutes, 10 seconds. I reckon that's good enough. So thank you, everybody, for listening. And I hope you can use this as a tool in future years to come and pull the bits out of it and prosper. Be your own plant pathologist. Not go to the big major stores and ask them for their help, which is often not the best because they only know products. So they'll just try and sell you a product. But you, with all this background information now, can go and make your own assessments and don't have to use fungicide, because you can just go and buy a little bit of copper and maybe mix that up in a 2% solution. That might be your fungicide if you have problems, as an example. So it's really good. Organics. I promote organics, and I hope you get into it too. It's hard to do at the start because it's just a lot to get your head around. So if you do synthetics, fair enough, mineralized fertilizer, but try and transition. That's why I hope that everybody can transition into fully organics and be sustainable. And let Mother Nature say thank you. I'm going to end on that note. Thanks everybody for listening or rewatching. Happy breeding, happy growing, and good health to you all. Bye-bye. See ya.